information already about the general foods, places to eat, or something like that. And there's a conference dinner tomorrow, and it's free food, so we'll have something for you to sign up here. So anyway, it's very good to have you here. And our first speaker is Professor Jennifer Schwarz from Syracuse University, and she's going to talk about using model active tissues containing active chromatin to test multi-scale hypotheses in organoids. Thank you, Jen.
Okay, thank you, Danilo. Yes, the title is a little bit of a mouthful, and I was thinking I was going to go at the end of the conference, so hence this sort of more mouthful of a title. But it's great, it's wonderful to also go at the beginning. So I'd like to just begin by showing you some images of things that you may not guess what they are, but given the title, you could perhaps surmise what they are. Oops, so okay, great. So on the left, we have many cells. In the middle, we have a cell, a pictorial representation, and on the right, we have a cell nucleus. So we're going to be talking just a few minutes about each of these. Given sort of the amount of content, please do not hesitate to reach out to me if you want to learn more. So before I begin, I'd like to give a big shout out to my collaborators, Tao Zhang, a professor at Shanghai Jiao Tong University in Shanghai, Sartik Gupta, a newly minted postdoc working in the group of Fred McIntosh at Rice University, Shabib Amin, another graduate student, Quan Liu, who was a former graduate student and now a postdoc, and Ed Bannigan, who's a postdoc at MIT, and Alison Pattison, who's an amazing experimentalist at Syracuse University. So whenever you see SU, that stands for Syracuse University, and a big shout out in particular to Tao for this talk, and then also to Sartik. Okay, so. And also, I would like to acknowledge some of my experimental collaborators, in addition to Ali Pattison at Syracuse University. So there are some other experimentalists I'd like to, that I'm collaborating with and like to acknowledge. Okay, so our brains are made of soft matter. Okay, so I'm at the right place, right? Lots of different types of brains. Here we have a scale bar. We certainly have an idea of our brain size and maybe the cat, the mouse, and whatnot. And here is a dolphin, which is a beautiful creature to study. And you might say when the high energy people, since we're near the high energy, or this institute has a lot of high energy theorists, and when we talk to the high energy theorists, they say, oh, well, you're doing low energy. And I say, well, yes, but the brain made of soft matter came up with the standard model, right? Or a bunch of brains collectively. And so, beat that. All right. And so now one can ask the question, given these different pictures, how do human brains differ from, say, gorilla brains or a mouse brain? And that, of course, people have been asking that question for centuries. We certainly know or are aware of, since people have been killing each other, know about sort of the folds and whatnot in our brains and the different shapes and sizes. And so how does that emerge, and what makes us different from, say, a chimp? And so that's a hard question, clearly. But let's ask a slightly simpler one. And the one is the following. Let's ask a question, the same question, but reframe it in the context of what is known as a brain organoid. So organoids are a collection of cells. You take what are called stem cells, which are unprogrammed. They don't know what type of cell they're supposed to be yet, a bone cell, a heart cell, a kidney cell, a neuron, and you grow them, these cells, so human pluripotent stem cells is that acronym there. You grow them in some sort of protocol that the biologists have uncovered, namely biologists such as Madeline Lancaster, who's really the brain organoid pioneer, along with Noblick. And so this is the first brain organoid paper, really. There might be some might dispute that, but in 2013. And here is a cross-sectional image of a brain organoid after so many days in development. Notice the scale bar, 200 micron scale bar. the colors are staining. So Hirsch stain is staining for nuclei. The other stains are staining for other things. But notice that the very kind of contorted shape and whatnot. And so here is a brain organoid. And so Madeline asked, oh, and I should say the brain organoids are very interesting in the sense of after you grow them for now 3, 4, 5, 6 months, you can keep them alive for many months. They're kind of small. They're like little tiny raisins because they don't have vasculature. They don't have a circulatory network to deliver oxygen to the cells in the core. So they're limited in size. Of course, people are obviously working on that in terms of incorporating blood vessels into all sorts of organoids. But the brain organoid in particular, because it's brain as opposed to kidney, the cells, these stem cells, eventually develop or become, some fraction of them become neurons, and those neurons do fire electrical signals. So this is a cross-section of one of these slices of the brain organoid, and then this color bar denotes spike activity. So there actually, you can, if you will, attach electrodes and record the neural network activity in these brain organoids. And here, anyone here I've become now a computational neuroscientist this past year. Here, sort of spikes of these neurons firing. And also, the brain organoids, you saw that initial shape that looked very contorted. But of course, not of course, but what happened in the lab of Orly Reiner, if you grow brain organoids in a quasi-2D confinement, so between a membrane and a cover slip of about 150 microns, they become sort of flattened, and this is the top view, and then you see, aha, maybe this looks a little less contorted. It looks more like a brain, where these lobes, if you will, are like the folds in the brain that we have. And so there's all sorts of interesting things one can do. And so what Madeline did is she asked the question, when we go back to human versus chimp or human versus dolphin or different types of brains, you can ask, well, do human brain organoids evolve differently than, say, chimp brain organoids? So now we replace brain with brain organoid. It's in a petri dish. It's simpler to sort of, in principle, to understand, though complex as well. And so when Madeline asked that question, so she took, if you will, stem cells from a human, stem cells from a chimp, stem cells from a gorilla, and then grew these brain organoids according to her now modified protocols and whatnot. And here is images of, in terms of day 3, day 5, day 10 going across. And going from up to down is human, gorilla, chimp. Again, this 200 micron scale bar. And what Madeline and her group found is that if you look at day 10, let's just look at the left bar chart, the organoid area for a human is a bit larger, at least almost 2 times as large as the chimp and the gorilla. And so we know that human brains are larger actually than chimp brains. And so here we also see in this setting of organoids that the gorilla and the chimp are smaller than the human brain organoid. And there's a lot of beautiful work in this paper here that came out in Cell in 2021, where Madeline and her group really tried to figure out what is going on, why the difference in sizes. And in the end, certainly biologists, what they will do is they will look for differences in gene expression. So gene, you have your DNA, part of the DNA that codes for some protein, or maybe doesn't code for some protein. Gene, the Greek word is, I guess, birth, to generate. So that's sort of your measure of inheritance. And so they found this sort of interesting difference, or this increase in expression of ZEB2, which is a particular gene, a transcription factor, as one calls it, that regulates transcription in terms of a difference in expression between the human and timing in terms of the human versus, say, the gorilla here. This is a picture of a gorilla. So there's a lot of biology there. And as a physicist, you might say, well, that's wonderful. Oops. And then you could ask, well, this is what biologists are asking all over the world. How do genes affect tissue scale or organoid scale organization, and vice versa? Because there's feedback between the two. And this is obviously a very hard question, and one that obviously we need to eventually get at. And so where do we start? how can we start with this problem? As a theoretical physicist who's studied lots of different things in terms of disordered systems, phase transitions, rigidity percolation, and whatnot, now I've become more lured into this field of biology, asking these tough questions, but trying to build minimal models to try to at least maybe not answer this precise question with so much detail, but to get sort of underlying principles, physical principles and mechanisms to understand how at the smaller scale, at the now we'll call chromatin scale, can affect tissue scale organization and vice versa. So obviously here is a picture of a cell here, a mouse embryonic fibroblast from Alison Pattison's lab. Here is this nucleus in here, denoted by red. And when you sort of zoom in and look at the structure of a cell nucleus, here is a schematic. You have your DNA. This has your base pairs, and then you have these histones, so the DNA wraps around because you have at least human DNA about 1 meter, and how does it actually go into this, if you will, 5 micron-ish size nucleus. You have these spools, if you will, of histones, and then they wrap around, so the DNA wraps around histones. You form then chromatin, so that's sort of the buzzword. It's these DNA plus proteins. And then you have higher, sort of more multi-tiered structure in terms of chromosomal territories and whatnot, but that's all terminology. So for us, in terms of thinking about a simple model for a nucleus, here is sort of a nice, again, slightly simpler drawing in terms of a cross-section of a cell nucleus. And interestingly, there is in here the red. It's called the nuclear lamina. So you don't just have what I'll say now chromatin, and then the inner and outer nuclear membranes, which is what I used to think in terms of my 10th grade biology. That's the last time I had biology, a long time ago. But there is a lamina shell, which is made up of some filamentous proteins, like a fibrous cage that helps protect, if you will, the chromatin. Now, of course, that was a drawing, and you say, well, Jen, and as a physicist, you might say, well, a cell nucleus, the shape is, a spherical cow. That's the shape we should assume. Of course, when you start looking at actual images of here is a cell nucleus, just staining the nucleus, you see that doesn't look like a nice sphere, which was depicted in the pictures on the previous slide. And this is, if you will, a healthy cell, and it gets even more crazy when you have disease cells and the nuclei in those cells that contain this chromatin, this genetic information, can, in principle, depending on the type of disease, become even more sort of distorted in shape, at least distorted from your simple spherical shape. And so the question would be now a simpler question as opposed to that bigger multi-scale question, what biophysical mechanisms drive cell nuclear shape? And so here is a simple minimal mechanical model for a cell nucleus. This is a cross-section. We have here in the purple denotes the lamina shell. It's a really simple bead spring model. So you see these beads, there are springs between them. So this lamina shell I mentioned, it's treated as an elastic, deformable shell. You also have chromatin, which is, for this particular model, one long chain. You also have linkages between the chain and the shell. So they are coupled. You also have chromatin cross-linkers that are present. There's a whole world of rheology and mechanics of cell nuclei. You have these cross-links that help give the cell nuclei some stiffness. And you also have motors like RNA polymerase II that transcribe, or motors such as cadensin that actually sort of form loops and whatnot and remodel the chromatin. And so there's lots of things happening. So here we can have different types of motors. We can have extensile motors. We can have contractile, at least in the model, contractile motors. Now how these motors technically usually work at a much smaller scale, how they manifest themselves at a chromatin level, there's some hand-waviness. But anyway, this is what we would call activity. So now we come to this sort of active matter. And then we put this all in a model, and the motors make the chain active. And here is the 3D version where the purple is the lamina shell, so we have the same coloring. And now here we can say, we have this computer simulation. Let's let it rip. Let's study it for certain parameters of the model. Let's look at its shape. of the overall nucleus. Let's look at what's going on inside. This is quite, maybe an even complicated model, but most people that study chromatin actually really think of they use a fixed shell, and then they have chains moving inside. But in my mind, the coupling of the shell, the deformable shell, to the chain is really key. And so I'll just very briefly say, how can we calibrate this model? Does it have any bearing on reality, right? Because as a model builder, okay, you can do whatever you want, but now we need to be constrained. And so just to go quickly, in terms of chromatin correlated dynamics, this was discovered by Alexandra Zitskova and others in 2013. Here is a cross-section of a cell nucleus where the histone motion has been tracked. Those little, if you will, spools upon which the DNA wraps around. So the histone movement has been tracked, and the different colors denote different direction. So either north, south, east, or west. And the histones are followed over a time scale of about 1/4 of a second. Now, as you go to the right, the histone's motion is tracked for 2.5 seconds, and then for now 5 seconds. And notice that as you go tracking and now go to 10 seconds, here you notice that the colors start to merge, meaning there's regions of the chromatin that move in the same direction. Maybe you could think of this as flocking or something, maybe slightly more complicated than that. It's only over a certain time scale. As you go to larger time differences, I should say, then there's decorrelations involved. So this is a 2D representation of what's called correlated chromatin motion. And here is a one-dimensional representation of that from the experimental data, where what is plotted is the spatial autocorrelation function. So you ask, here's a piece of chromatin, here's a histone some distance away, delta R, and you're asking if they're going in the same direction, which would be 1, if they're going opposite direction, -1, and if they're randomly moving in a direction random with respect to each other, it's 0. And so this is a plot of this function as a function of delta R, so asking over what length scale do these correlations happen, and the different colors are for different time windows. So notice that this 10 seconds of this bright green triangle, that's where you have the largest correlation, even out to delta R about 6, and we're talking almost to here's the delta R across the full nucleus. And so granted, the correlation is not 1, it's not 0.8, it's around 0.2 out here, but still, that's some significant correlation that one didn't expect. And I will just say that the model that we've built, Kwan, Ali, and Ed and I, if we measure those same correlation functions for a rigid shell versus a soft shell, we do see these same types of correlations. The gray is for no motors. The blue is for extensile motors, and again, different time windows as you go from light to dark. And the red is for contractile motors. So for these two different types of motors, we see for the soft shell case significant correlations, obviously clearly more significant than the experiment. But the point is that because this chromatin is really in a liquid state, you might say, how do we get correlations then? Well, 1, we can cross-link. That will help get correlated motion. 2, because there are links to the shell is elastic, and so you can have indirect correlations, if you will, driven through the mediated interactions through the shell that help generate more longer range interactions in the chromatin, even though they're not explicitly in there. And so we can also determine and find wrinkles and bulges, obviously with this deformable shell, and that's another type of calibration in the sense of, do we see, what types of shapes do we see in the nucleus? And so you can sort of do the same thing one does in cardiography, making maps. You can say, make a surface map of the cell nucleus, and then I do a stereographic projection. And so here is the stereographic projection for Earth. Here is the stereographic projection for the cell nuclei, and you can identify the hills and the valleys, or the bulges and the wrinkles, as we like to say. And interestingly, with this particular model, if you decrease or change the number of motors, so this is the number of motors, and this is the number of now bulges, so the outdents, if you will, not the indents, and for different numbers of crosslinks, that the number of bulges increases with the number of motors. And you could say, okay, Jen, well, that's a quantitative result, whatever. But interestingly, how does that correlate or translate to experiments? Interestingly, so this is work done in Drew Stevens' lab, where actually if he inhibits motors, so decreases the number of motors by decreasing transcription or inhibiting transcription, the blebbing, so I guess some people call it blebs, we're calling it bulges, decreases. So, or the percentage of cells that bleb do decrease when you, if you will, decrease the number of motors, just as we see in our numerics. Now, again, right now there's not purely quantitative agreement with the model, so that's what we're working towards in terms of what types of details should be incorporated. And so you can then also ask what's causing these bulges in terms of this biophysical mechanism generating weird nuclear shapes. Well, the claim could be a conjecture, this correlated chromatin motion, so you have chunks of chromatin that are moving in certain directions in a correlated way over some time scale, that those bulges, if you will, are generated by the chromatin pushing, because they're excluded volume interactions as well, pushing on the shell. And so correlated chromatin motion could be a mechanism for generating bulges. So in the interest of time, I'm going to go through this very quickly. Now we say, oh, Jen, you've been talking about the cell nucleus again and chromatin organization. How do we get to organoid scale organization? So that means that we need to build a model for an organoid. So in summary, this is a bulk model for many cells. So each polyhedron represents a cell. There's a certain energy functional in terms of there's a volume spring, so cells prefer a particular volume. Cells also prefer a particular surface area spring, given the quadratic pieces here. And you can, of course, the vertex, this is a vertex model, a 3D vertex model, starting back to the days of Honda and Iguchi and others that have really done beautiful work. And what you can do then is ask about building this model, this 3D vertex model. How do cells rearrange? How do they move? And so it's quite, there's some interesting work in terms of done by S. Okuda, and as well as actually earlier work in trying to figure out if cells, if you have a space-filling packing, if you will, so no gaps between the cells, how do cells rearrange? And people have thought about this even way back when in terms of metallic grains and whatnot. So I think I'm going to skip all this in the sense of there is rigidity transition in this, and maybe our plenary speaker will talk more about that. If you are interested in simulating many cells, there's a 3D vertex model code that Tao Zhang and I have developed with Tao doing the actual writing. And there is a GitHub page for that. You can certainly download it. I will show you one interesting movie where you can ask about the rheology of this clump of cells, this organoid now, as you stretch it, as you pull it, and do lots of things. So lots of measurements now are being done on the model. And of course, we'd like to do, and I'll skip this, we'd like to do the same thing, obviously, with the organoid. So now that we have a cell nucleus model and a organoid cellular level, multicellular model, now we're ready to start to ask this multi-scale question where we now embed, say in certain cells, we embed a nucleus inside a cell. And all I'll say for now is that as you contort, say, the organoid by compressing it or stretching it or cells growing that help contort it, you have a distortion of the cell shape. And so people have shown over a number of years that as you change the cell shape, that the nuclear shape tracks the cell shape. So you can have changes in cell shape. And so then that leads to changes in cell shape, lead to changes in nuclear shape. And so you can squash the nucleus. And then as you ask about what happens as you squash the nucleus, okay, it's still running, sorry. So that's the nucleus being squashed, not symmetrically, asymmetrically, my apologies. You can ask about how the chromatin moves as the deformable shell is changing shape. And so you can track the differences and say, if I have a slight perturbation in the organization of the chromatin, and now we start to latch onto perturbation, meaning human versus chimp. If there are perturbations in the chromatin organization, how do those small perturbations in organization change when the cell nucleus gets squashed? We can start to sort of address these multi-scale questions. And here is the first, and now I'll end. Here is the first multi-scale kind of hypothesis of its kind in trying to really get at why human brain organoids now are bigger, if you will, than the chimp brain organoids. So I'll leave you with that. Please, there was a bit of maybe too much content, but please come and ask me questions. Thank you.
Thank you, Jen. I have a quick question before. In the mechanical model for the nucleus, you had those connections between different elements. Do you model them like elastic bands? Or
Yeah,
are
springs,
they like,
that's springs. But of course, there are viscoelastic, meaning there's presumably some time scale, or can be rupture, so above some strain, critical strain in that cross-linking spring, it could break. So for now, we haven't done that because we're looking at time scales, really, that simulation time window of that blob thing, it was of order 10 to 20 seconds. So, then the question is, what's the time scale for, what's the time scale between ruptures? That is being, experimentalists are trying to really measure that now. So,
okay.
yeah, great question.
More questions?
But along a similar line, so is this simulation just a molecular dynamic simulation, or is it more complicated?
Yeah, it's overdamped dynamics.
It's overdamped? Okay.
Overdamped dynamics.
Overdamped dynamics.
No hydrodynamics, no inertia.
But then fluctuations.
And there are some fluctuations we would call, active fluctuations, if you will, due to motors and all sorts of processes that are going on in the cell.
Okay.
So apologies for not mentioning that.
Thanks for this very nice talk. In the human body, or any body, we have different types of cells. Can you trigger or adjust this model for describing these other types?
Bingo, yes. So clearly, I'm focusing on brain organoid just because, well, Madeline is amazing, and also I'm learning about neuroscience and whatnot. But clearly, yes, at least this vertex model, in principle, could be applied to any type of cell. The issue becomes that very simple energy functional in terms of the area spring and the volume spring. As cells evolve, I think more terms, if you will, they develop polarity, all sorts of complicated inner machinery. That energy functional, I think, is not unreasonable as a starting point maybe for baby cells or something, but then as they sort of start to recognize who they are, then that becomes harder. And another issue is with, say, neurons. They're very long and skinny with the axons. So, then the question becomes, this vertex model maybe is not as good. So there are limitations to this modeling, but in principle, you change parameters, you change energy functional, you could, in principle, describe different cell types.
Suppose you have a gel confined in some space, some region, and then you have all those cross links and maybe even constant springs inside. And if you push it, no friction. Would you have this, no motors? Would this pushing with no friction will have some behavior similar to this, of these bundles, this thing, this...
the pulling of
yeah.
That's a great question. So I went over it quickly. So there is this parameter, a shape index parameter for the cells, which is a dimensionless parameter, which is in 3 dimensions, it's the area divided by volume to the 2/3. So that gives you some idea of the shape of the cell. If you're a cube, you have shape index of 6 and whatnot, so different numbers correspond to different shapes. So if you change that parameter, that's an S0, that's, if you will, the equilibrium surface area of spring constant. If you change that, you can toggle between a fluid-like system of cells and a solid-like system of cells. So if a gel is more like an elastic type system, if you did that pulling, I would suspect indeed exactly that clump of cells, that organoid, biologists don't like it when I say clump of cells, that would be more like a gel pulling on it. That's right. However, with this model, you can go to a fluid phase, and then it sort of, as you pull it, it'd be more like, more like a viscous medium. Okay. So you can, viscoelastic maybe more generally, but indeed there's certain regimes where it'd be like a gel pulling. Exactly.
Okay.
Yeah, great observation.
You.
Can you use your model to model cardiomyocytes or muscle cells or things like that?
Muscle cells?
Yeah.
In principle, I want to say in principle, yes, but I haven't actually read the details of muscle that's striated or something, and so yes, we'd have to have sort of elongated cells, maybe in certain directions and pattern, and maybe we think of pneumatics or I don't know. So yes, in principle, yes, but I haven't thought precisely how to do it yet. But you've now motivated me to think more about that.
Okay, one last question by Ron. Pablo.
Some forms of active matter models are characterized by giant number fluctuations.
Yes.
Do you see evidence of giant fluctuation in this?
That's a great question. Yeah, actually, those giant number fluctuations. Yeah, I haven't studied that precisely. Density fluctuations have been studied in this model, and actually we see hyper-uniform type density fluctuations, broad spectrum density fluctuations, but I have not studied that particular quantity. That's a great point, because yeah, these vibrated layers, vibrated surfaces of rods, and beautiful stuff there. So that's a great point. I'm thinking about that too.
Okay, let's thank Jen again. And now I invite our next speaker, Professor Luis Roberto Evangelista.